This is the lecture for Lab 3, Introduction to the Microscope. The purpose of this lab is to get you familiar with using a microscope, and that will include learning the parts of the microscope, learning how to correctly clean the microscope, learning how to correctly focus the microscope, and learning how to use oil immersion. The first thing you need to know are all the different parts of the microscope. So starting with the ocular lenses are called ocular because they're close to your eye. Then moving down from that, this is the head of the microscope. Then below that is the nose piece, and the nose piece has the objective lenses. And the objective lenses are called objective because they are close to the object that you will be looking at. This is where you place the slide. Then this is the stage. Right under the stage is a condenser. The condenser condenses the light, and there is a knob which allows you to move it up and down. Below that is the iris diaphragm, and there is a little lever where you can open and close it, and that controls how much light goes through the slide. Then, of course, there is a light source and the base. Going on the other side, this is the arm. So this big support is the arm, and that's what you carry the microscope by. Then on top of the stage, you have stage clips. They hold the slide on the stage. Then you have the fine adjustment knob, which is the smaller one here, and the coarse adjustment knob, which is the larger one there. And then there's the mechanical stage adjustment knob that moves the slide. And then there's the light source adjustment knob, and that is a dimmer, basically, controlling how much light is going into the microscope. I want to go through some of the parts of the microscope in a little bit more detail. And the first part I want to talk about are the oculars. Remember, the oculars are close to your eye. So these are binocular microscopes, bi meaning two, because there are two ocular lenses. And these ocular lenses magnify by 10 times. And the ocular lenses, you can adjust them. They can slide around this knob, so you can increase or decrease how far apart they are from each other. And this is very important because you want to adjust them for the width of your eyes. Everybody's eyes are a little bit different lengths apart, so the width between your two eyes is a little bit different. So when you get your microscope, you want to adjust it to fit the width of your eyes so that you can look through both oculars at the same time and comfortably see one field of view. It'll be one circle of light. The next part of the microscope I want to talk about is the nose piece. So this is the nose piece, and on the end of the nose piece are the objective lenses. And these objective lenses, you have 4 objective lenses on our microscopes, and this part can rotate around. So you can choose which objective is over the slide. The slide would be down here. And our microscopes have 4 different objectives, and this is the power by which they magnify. The shortest one, the lowest magnification, is a 4 times objective. So it magnifies 4 times the normal size. And we call this the scanning objective. And we call this the scanning objective because it has the biggest field of view, so you can see the largest area of the slide. So when you are using the 4 times objective, you're looking through the oculars, which magnify by 10 times, and you're using the 4 times objective, so you multiply by 4. So then your total magnification is 40 times. And we put a little X, meaning times. So when you're under scanning, looking at the scanning objective, looking through the scanning objective, you are magnifying the objects 40 times. The next higher power is 10 times. So when you use the 10 times objective, your total magnification is 100 times. Then the next one magnifies by 40 times, and I often call this the high and dry. High and dry objective. So high and dry because it is the highest magnification that doesn't use oil. Then the highest magnification or the highest objective you have is the oil immersion lens. And I will call that the oil lens, or I'll say look at it under oil. And this magnifies by 100. So of course you multiply by the objective, so your total magnification when you use oil immersion is 1,000 times. The next part I want to talk about is the stage. So the stage, this is the stage. It is right below the nose piece, and on the stage you have the stage clip. So the stage clip here, it holds the slide. So you'll place the slide, it should fit perfectly right in here. And there are two knobs I'll show you in a minute, which you can use to move the stage clip back and forth and also up and down. So you don't need to use your fingers to move the slide. So once you put the slide in, you would take the slide, place it into the slide clip, then you want to position it so the colored part of the slide, your stain, is right over where the light comes in. So you want to make sure that whatever area you want to observe on the slide is placed right over the light. Next, I want to talk about some parts of the microscope that are under the stage. So this is the stage, and below the stage you have this knob over here, and it has two parts. One part will move the stage forward and back. The other part will move the stage to the right and to the left. So that is the mechanical stage adjustment knob. Then below the stage you will have the iris diaphragm, and on the iris diaphragm there's a knob somewhere that you can slide back and forth to open and close the iris. Below that is the condenser, and on the other side of the microscope there should be a knob where you can raise and lower the condenser. And then below that, of course, is the light over here. And then on one side you will have the light switch to turn the light on and off, and there is also a dimmer switch. So you can increase or decrease light intensity that way too. The last part of the microscope I want to talk about are the knobs you use to focus the microscope. And on both sides of the microscope you have this set of knobs. The outer larger one is the coarse focus knob. The smaller inner one is the fine focus knob. When you are focusing the microscope, you always start with the lowest magnification objective. So you start with the scanning objective. So this is the scanning objective here. Then you place the slide into the slide clip, and remember you place the object you want to look at over the light. When you are focusing, you are going to use the coarse knob first. And the coarse knob is physically going to move the stage up and down. So you are going to physically move the stage closer or further away from the objective lens. And normally you start with the stage all the way up, so you slowly move the stage down until the object comes into focus. So once you have used the coarse focus knob to bring the slide into focus, then you fine-tune your focus with the fine adjustment knob. So you just fine-tune the focus, and the fine adjustment knob does not move the stage up and down. Once you have perfectly focused what you are looking at using the scanning objective, the 4x objective, then you switch objectives to the next highest power, so move the nose piece so that the 10x objective is over the slide. And when you do that, you only need to use the fine focus knob. So these microscopes are called parfocal. That means they are adjusted, so when something is in focus with the scanning, it should be pretty close to focus with the next power up. So you do not move the stage after you have used the scanning objective. So you switch to the next higher power, the 10x. You use the fine focus to fine-tune your focus, make it come in clear. Then you switch, move the nose piece so the next higher power comes up. This would be the high and dry. And again, just use the fine focus. Then the last one, you will use the oil. I'll talk about specifically how to do oil immersion in a little bit. But again, as you switch to the oil immersion lens, you are only going to use the fine focus. So it's really important to note that you only use coarse focus when you are initially focusing the scanning objective, the lowest objective. If when you have moved to one of the higher objectives, the 40 times or the 100 times, notice that these objective lenses are physically longer than the scanning one. And if you actually use the coarse focus and move the stage up, you risk breaking the slide. So that's why it's really, really important to note that you only use the coarse focus when you are focusing on the lowest power. After that, you never use the coarse focus again. The microscope is parafocal, so everything should be in pretty close focus. You only need to adjust the fine focus. Because bacterial cells are so small, we need to use oil immersion to get enough resolution to see them clearly. So because we are using oil immersion, that means the microscopes need to have that oil cleaned off. And whenever you come into the lab and are going to begin using the microscope, you need to clean it right away, in case students who use the microscope before you have left oil on the lenses. In order to clean the microscope, you need optical lens paper, and the optical lens paper is a very light sort of tissue paper looking paper. You will also need lens cleaner. Lens cleaner, it's either a blue or purple kind of liquid in a squeeze bottle. And when you are cleaning the microscope, you are going to focus on the lenses on the objectives. So this is the part of the microscope you are going to be cleaning. Now with the 4 objectives, the oil objective, of course, should be used with oil, so for sure this will be dirty and it will have oil on it. So for sure you will need to clean the lens on the oil immersion objective. And often you want to clean the high and dry. We call it high and dry because you should not be using oil with it, but because it is a similar length. And if a student has gone back and forth between high and dry and oil, it often gets oil on it. So it's probably just good practice when you come into the lab is to automatically clean the oil lens and the high and dry lens. So you take a piece of lens paper, put a couple of drops of lens cleaner on it, and just rub it over the lens. You don't have to worry about scratching the lens because this paper is specially made to not scratch the lenses. Another thing you may want to pay attention to is if a student is very messy and uses too much oil, oil might get on the stage. And if oil has gotten on the stage, then you need to use the lens paper and the lens cleaner to clean the stage also. Using the microscope. Before you do anything with the microscope, you want to clean the microscope. So like I mentioned before, use the optical lens paper and the lens cleaner to clean the oil immersion objective for sure and the high and dry objective. Once you have done that, then you are ready to use the microscope. So the first thing you are going to do is adjust the oculars so that they fit your eyes. So again, you can slide those oculars kind of up and down around this pivot. And what you want to do is when you look through the microscope with both eyes open, you want to see one circle of light, and this is your field of view. So you want to first adjust that. Then keep in mind when you're looking at the field of view that at the low magnification, so using the 40 time objective, you are going to have the largest field of view. So the largest field of view, and as you increase magnification, your field of view is going to get smaller and smaller. So whatever object you want to look at, you want to make sure it is centered. Because if the object is over to the side, then when you get a smaller field of view, that object may be outside your field of view. So always use those stage adjustment knobs to keep whatever object you're looking at in the center so that you know as your field of view gets smaller, you will still be able to see that object. Once you have cleaned the microscope, adjusted the oculars so that you see one field of view, then you put your image, your slide on the microscope. And this is just a slide of a root tip. But when you place the slide, you will be using the lowest magnification, and our lowest magnification will be 4 times objective. And what you want to do again is center what you're looking at. So you want to make sure this part of the slide is in the center of your field of view. And when you are focusing at the scanning objective, use the coarse focus knob to move the stage down until you can see clearly the object you're looking for, and then you use the fine focus to fine tune it. Once you have done that, you switch to the next higher objective. So your field of view gets smaller and the cells get larger, and then you use only the fine focus. And you continue doing that sequentially. This image has more lenses than what we use, but you switch to the next higher power, use the fine focus, next higher power, use the fine focus, next higher power, use the fine focus, and so on, till you get to the last objective using just the fine focus. And again, make sure whatever you're looking at is in the center, because if this area is in the center, then pretty soon this would be the area you're looking at and you wouldn't see any cells. Also, another thing is make sure you focus everything perfectly so you can see everything clearly before you move on to the next power. Because if you don't see it clearly here, you will not be able to see it clearly later on. And then another example of this, these are human red blood cells, and you can see that, of course, the magnification is getting higher and higher and higher with each objective. So the cells are getting larger and larger, and your field of view is getting smaller. Eukaryotic cells are very large, so using a 40 times objective is enough magnification to see them pretty well. But prokaryotic cells are much smaller than eukaryotic cells, so we need to use a higher magnification. And the only problem with a higher magnification when we use 100 times objective is that we're going to get an increase in refraction of light. And refraction of light is the bending of light. So this is a diagram of a microscope. This is the light source down here. And the light is going to have to pass through the condenser lens, the iris and the condenser lens. Then it is going to go through air. It's going to go through the slide, air again, before it gets to the objective lens. And every time light changes medium, when it goes from a lens to air to a lens, it bends. So when you use a higher magnification, you're going to have more light bending and not passing through the microscope. So it's going to make it harder to see the image. In order to prevent this refraction, this bending of light, we use immersion oil that has the same density as the glass and the objective lens, so that keeps the light moving towards your eye. So using immersion oil reduces the refraction of light, which improves resolution. And again, resolution is the ability to distinguish between 2 points. So because we're using such a high magnification for the prokaryotic cells, we need to use immersion oil to prevent that refraction. The immersion oil you are going to be using is in a small vial, and in this vial there's a little glass rod that extends from the lid. And so when you take the lid off, this rod is going to have oil that drips down and drips 1 drop at a time. So in order to do oil immersion, what you are going to do is use the microscope like you do before. Start on the scanning, go to the 10 times, go to the high and dry. And once you can see the microbes clearly on the high and dry, you will shift the high and dry objective over so that you have an open space over your slide, and then you are going to use this glass rod to drop 1 drop of immersion oil right over the light coming through the slide. Once you have dropped 1 drop of immersion oil over where the light is coming through the slide, then you will gently slide the oil immersion lens over, and that will create a continuum between the oil immersion, the oil and the slide and the lens. I just want to go over some common problems that come up with a lot of students as they're learning to use a microscope. First problem is that the image is too dark. So it's way too dark. For this, you need to check all the ways you can control the light. First thing, check the dimmer. Often the dimmer is turned too low, and so not enough light is going through. Then you want to check the iris. So the iris diaphragm to make sure that is open enough, and also the condenser. So check all of those things. All the microscopes are checked before the semester, so we know they are all in working order. So make sure it's not something you need to adjust to fix the light issue. Then number 2, there's a spot in my viewing field. Even when I move the slide, the spot stays in the same place. This is often because your lens is dirty. It could be the objective lens or it could be the ocular lens. So you want to use the lens paper and the lens cleaner to clean the lens. And sometimes you see things that look like that or long fibers. We call those artifacts. And those are just things that they're not cells, they're not what you want to look for. The number 3, I can't see anything under high power. And I mentioned before, if you can't focus under the scanning or lower powers, then you're not going to be able to see anything under high power. So make sure you can see everything clearly with each objective before you move on to the next one. And the last one, sometimes you see this. Only half of your viewing field is lit, so it looks like there's a half moon. That means your objective wasn't fully clicked into place. Or if you see that it's all black, your objective still is off. It hasn't been clicked into place. So make sure you check your objectives if you see something that looks like this. The most common problem with microscopes are having dirty, and when I mean dirty, I mean oily microscopes. This is an incredibly common problem. And it's because we are using oil immersion and we have many students using all the microscopes and they are not cleaning them appropriately. So when you look at the bacteria, if the microscope is clean, you've done everything correctly, it should look like this. You should be able to see nice, distinct shapes of bacteria. They should have nice, clear outlines, and these are bacilli. Often what happens if the microscope is not cleaned well enough is that the image will be cloudy. It'll be fuzzy, and it can even be this bad. If you see this, that means everything you are doing is correct. The light is moving through. You are using lenses. You can see through the lenses. But if it's blurry and using the fine focus, you cannot get it clear. That means your microscope is dirty. And the only way to fix that is to clean it again. So that's why it's really, really important to just clean the microscope before you start using it, and also very important to clean the microscope when you are done, so you leave it clean for the next student. And this is just another example of a dirty microscope with the same image. This is what the image should look like, and if it's fuzzy, if you can't get good resolution, it is because your microscope is dirty, it's oily.